over there, the hypothalamus. So in your hypothalamus, if you were to draw a hypothalamus, okay, in the hypothalamus is there on both sides. So in one side of it, this is the periventricular. The periventricular nucleus of hypothalamus. From there, what is going to be released? Your CRH is going to be released. Corticotropin releasing hormone. Now, what decides whether CRH is released or not? How is it released? Okay, so one of the major things that affects this is impulses that come from your suprachiasmatic nucleus. Okay, that is also a part of hypothalamus. Suprachiasmatic nucleus is a regulator of your circadian rhythm. Okay, so your circadian rhythm is pretty important in maintaining your HPX. How so? Because this is not a continuous signal. This signal that is going to come from this, based on this, is going to be pulsatile in nature. And usually in our daytime, around 4 a.m. in the morning, that is when it starts to increase. And then again, before your regular meal time. So this is provided you eat regularly at a particular time. Okay, so say you eat breakfast at 8 o'clock, maybe a little before 8 o'clock, maybe from 7 o'clock onwards, glucocorticoids will be released more. Okay, and also this is just a starting point. When we talk about the peak, usually around 6 a.m. it peaks. Okay, so the peak comes to around 6 a.m. and the nadir, that is the lowest, goes to in around your midnight. Okay, so this is one signal that regulates your HP axis, your circadian rhythm. Now, beyond circadian rhythm, we have emotions. Okay, so from your limbic system, any sort of emotions, including any sort of stressor. It can be psychological or it can be physical also. So that is detected, one, by nociceptors pain, for example. Okay, or any other through the nucleus tractus solitarius, any other sort of efferent that comes. Okay, or even hypoglycemia. So all of these sort of stressors can also, along with emotions, stimulate the release of corticotropin releasing hormone. Now, beyond this, even pro-inflammatory cytokines. Okay, so the usual pro-inflammatory cytokines include your TNF. Then we have the interleukins, majorly 1, 2, 6. Okay, and there's another one which is leukemia inhibitory factor. Okay, and all of this also can stimulate your hypothalamus to release corticotropin releasing hormone. So what do you understand? Your hypothalamus from the periventricular regions release corticotropin releasing hormone. Now that is not a continuous signal, that is a pulsatile signal. Circadian rhythm is something that contributes to this signals that are coming from the suprachiasmatic nucleus. Okay, usually this peaks around 6 a.m. Around midnight, it is the lowest. On top of that, emotions, on top of the stresses, that is either through pain or any other sort of efference or even internal signals like hypoglycemia. And other internal signals like pro-inflammatory cytokines which are released. They can also stimulate the release of corticotropin releasing hormone. Okay. So now that corticotropin releasing hormone is done, it now has to go and act at the level of your anterior pituitary. So we'll just draw your anterior pituitary over here. Now, corticotropin releasing hormone, if you remember the previous lecture I told you, it is a type of a peptide. Peptide hormones generally act on membrane receptors. The type of membrane receptor that is involved here is G alpha S. So G alpha S, and like that, it goes to the anterior pituitary. Now, once it acts, there is a gene which is predominantly for a substance called pro-opiomelanocortin. So this is the substance that is produced, which is pro-opiomelanocortin. Okay, so we have pro-opiomelanocortin. Now this is a large substance. So one of the things that happens here is this large substance, which is your pro-opiomelanocortin. It gets split into various components by the effect of your corticotropin releasing hormone. So in this process, you don't have to know that much in detail. It activates the type of convertase which splits it. So what does it split it into? One thing it splits it is as it cuts, one part of it becomes what we call a gamma MSH, melanocyte stimulating hormone. And then another part over here, this becomes your ACTH. And then beyond this, we have another component, which is your beta lipotropin. Now, this beta lipotropin again will be split into various components. For example, gamma lipotropin, your beta MSH, and your beta endorphin. Now, this is the important part. Your ACTH, this was a small part that came out. This ACTH is actually made up of two segments. One segment is your alpha MSH. The other segment is what you call clip. Okay. So this is your corticotrope inside your anterior pituitary. So in the anterior pituitary, this is your corticotrope, from where your ACTH is finally released. Okay, so we'll talk about this importance of this MSH and all that later. So just understand this for now. So again, what is happening? CRH is released. It acts on the anterior pituitary and it splits pro-opiomelanocortin. Two different components. One among that is ACTH. ACTH also has an MSH component, also has a clip component. Now this ACTH is being released down. Okay, I hope you're with me till now. If you have any doubts, please do let me know. Now, from here, this is gonna go and act on your medulla. Sorry, not medulla, adrenal. My bad. So in the adrenal, we have the medulla. And beyond that, we have the cortex. And that cortex can actually be divided into various zones. We have the zona glomerulosa, zona fasciculata, zona reticularis. Okay, so this part over here, this is your zona fasciculata. So this is where your ACTH is predominantly gonna go and act. Okay, so we got your zona fasciculata over here. Beyond that, we have zona glomerulosa. And we have zona reticularis. And this is your medulla. Okay, now once ACTH acts, ACTH goes and combines with a melanocortin 2 receptor. So this goes and acts on a melanocortin 2 receptors, which results in formation of cortisol. So cortisol is a major glucocorticoid in our body. Now cortisol, once it is formed, this by itself has an inhibitory activity. One, it goes back and inhibits this ACTH release from here. Two, it can go back and inhibit this CRH release and activity. So it acts on two different segments. Okay, so this is sort of a negative feedback loop. I hope you're with me till now. So what is the importance of this? If cortisol is there, it is there adequately and it is produced, it is going to inhibit itself. Now, the problem arises when from outside we're going to give some external source of cortisol. Okay, so external glucocorticoids. So this is an example where we give steroid for therapy and external glucocorticoids are usually given. Or say he has any sort of hyperactivity of your adrenal cortex. So one example of that is your Cushing syndrome. So in both these instances, what is happening is, as long as a large enough dose is given and it is given for a long enough time. So usually if given for more than 1 week and a dose of more than 5 mg. So it depends actually. Okay. So based on this, if a large enough dose is given, this goes back and inhibits your CRH and ACTH. Thus, this whole axis is gone. So this is what we call HPA axis suppression. So why is this important clinically? So when you give external glucocorticoids for a long time, provided you give a large and long dose, usually it can take, especially if a large and long dose is given, it can take somewhere between 6 to 10 months even for recovery. So what is the problem? When you're giving external glucocorticoids, you suddenly stop it. Then, because of the HPA axis suppression which is happening, your cortisol will not be released. Cortisol is something that is required in our body, especially to respond to stress. So that HPA axis suppression makes sure that there is next 6 months also, there is no cortisol being produced. The body got used to an external glucocorticoid. So you should never stop external steroids just like that. Okay, it should be slowly tapered down. Okay, so this is one thing that I want you to understand. The next thing I want you to understand is there are two things. This hyperadrenalism, when I was talking about it, one, from the adrenal, increased cortisol can be produced for some reason. So usually anything that where increased cortisol production is there, we call it Cushing syndrome. Now, if specifically ACTH is more, either there is a tumor, most commonly it is some tumor in the pituitary, either there's a adenoma in the pituitary or some other external source of ACTH production is there. So ACTH being produced, especially because of an adenoma in the pituitary, that is what we call Cushing disease. So sometimes the question can come, what is the difference between Cushing syndrome and what is the Cushing disease? So this is the essential difference. One, the problem is ACTH production is increased. The other one, any reason why excess cortisol is produced, that is Cushing syndrome. Okay. So the difference here will be in Cushing syndrome, because cortisol is high, ACTH will be low. But in Cushing's disease, the primary problem is that ACTH is high. So ACTH also will be high, cortisol also will be high. So investigation wise, that you can differentiate. Now, another thing. ACTH has a component of alpha MSH. What you have to understand is this is the major one, MSH, melanocyte. Melanocyte is for your pigmentation, black pigmentation, melanin. So MSH is actually only 1 by 30 times as potent as the other beta MSH and all. But when ACTH is very high and then the concentration of this is high, then this becomes predominant and starts acting in the same way. So this can happen in two scenarios. Anywhere where ACTH is high. One, it can happen in your Cushing's disease, where a lot of ACTH is being produced. Two, it can happen in hypo. Hypo as in cortisol is not there. So when cortisol is not there, then this negative feedback on this is not happening. So what is the body's response? It increases CRH, it increases ACTH. Okay, so ACTH is increased in any sort of hypoadrenalism. One example of that is your Addison disease. Okay, so along with this only, we discussed a lot of clinical scenarios. Along with the further things that I cover, we'll slowly discuss more things. Okay, so we talked about Addison's, we talked about alpha MSH and why there is hyperpigmentation present in these scenarios. Then we talked about the negative feedback, HP axis suppression, the importance of tapering down glucocorticoids and all that. I hope you're there with me and you can understand whatever I covered so far. Okay, so if you have any doubt, please do let me know. I can see some students who are actually listening to the class. Okay, so now we'll come on to this cortisol. So again, in your zona fasciculata, how is this released? So cortisol is gonna be released somehow. Okay, so for that your ACTH is there. This ACTH goes and acts on your melanocortin 2 receptors. This is also a type of G protein coupled receptors and it is associated with G alpha S. G alpha S, what does it do? It activates adenine cyclase which increases your cyclic AMP, and that stimulates protein kinase A. So what does this protein kinase A do? So for that, let's go back and try to understand what type of hormone is this. We have peptide hormone, we have some sort of steroid hormone, okay, or amine derivatives. Can you try telling me what type of hormone is your cortisol or glucocorticoid? Yes, Pranali. Yes, so it's a steroid type of hormone. So steroid type of hormone, if you remember, it is not preformed and stored like your ACTH or a corticotropin releasing hormone, growth hormone, insulin, none of that. Okay, it's not preformed and stored, but when a signal from outside is coming in, at that time it starts synthesizing. Okay, so it's not preformed and stored in vesicles, but when an impulse like this ACTH is coming, at that time it starts the synthesis. How does it start its synthesis? For that, your common precursor for all of this is your cholesterol. The cholesterol is a common precursor for all of the steroid hormones that we know. Okay. Now, cholesterol, one thing from cholesterol ester, it needs to be converted by the activity of hormone sensitive lipase. Okay, so hormone sensitive lipase should act to convert cholesterol ester to cholesterol. This is done by this protein kinase A phosphorylation. Two, the LDL receptors which are put on the outside so that your cholesterol can enter inside. This is also done by this protein kinase. Okay, so one thing, the LDL mediated uptake. Two, hormone sensitive activity to convert cholesterol ester to cholesterol. That is done by your protein kinase. There's one more thing it does. That I'll talk about a little later. So because of this, what happens from here? This cholesterol is taken into your mitochondria. The mitochondria has an outer membrane and an inner membrane. So that is why there is a particular protein which is called star. So this helps move cholesterol from the outer to inner. So like this, cholesterol moves from the outer to inner. And then there is another component also, which is important, not only the mitochondria, but your sarcoplasmic endoplasmic reticulum. I'm sorry, it's not sarcoplasmic endoplasmic reticulum, the smooth endoplasmic reticulum. Okay, my slip of tongue. It is your smooth endoplasmic reticulum. So this cholesterol which reaches in the mitochondria, it gets converted to various stuff. And from there, it again goes to your smooth endoplasmic reticulum, from where it now goes back to your mitochondria. So shuttling like this happens. And finally, the end product of that is your cortisol. So what are the steps that are taking place over here? That is what we're trying to learn now. Okay, so you got this clear? Your ACTH is acting on melanocortin 2 receptors. G alpha S, so adenine cyclase, cyclic AMP, protein kinase. Okay, protein kinase, one LDL mediated reuptake, two hormone sensitive lipase activity converting to cholesterol. Okay, or it can also start a de novo synthesis, not only from outside, but from inside only it can start and produce a new cholesterol. Okay, the third thing your protein kinase A does is through CREB, which is just a transcription factor. You'll hear this very often in endocrine system. A transcription factor, just know that, which results in transcription of certain enzymes related to mitochondria, like CYP. Okay, so just know that this is the third thing protein kinase A does. So all of the things that are needed here, various enzymes are all needed here. So production of more of those enzymes, transcription and translation of more of those enzymes is also done by protein kinase A. So three things ACTH, when it acts, does, because of which cortisol is released. Now let's come to understand what is happening. Very simple. Okay, I'm not going to try to complicate. When you try to learn it by yourself and look at the diagram, it looks very complicated. But this is actually, I assure you, it's actually very simple. Okay, so again, we're going to start with your cholesterol. That is the starting point. We have your cholesterol. Now, this cholesterol is converted to pregnenolone. Pregnenolone is converted to your progesterone. Okay, so what is cholesterol to pregnenolone, pregnenolone to progesterone. Progesterone will be converted to 11 deoxy corticosterone. And from there, finally, your corticosterone is formed. So mind you, this is just a starting part. Okay. For this to happen, just remember some beta related enzymes are required. So over here, I'm just going to put 3 beta. Here, I'm going to put 21 beta. Okay. Here, I'll put a 11 beta. So just beta stuff over here. 3 beta, 21 beta, 11 beta. Okay. Now let's just add more layers to it. 3 beta hydroxy steroid dehydrogenase. Okay, so pregnenolone is converted to progesterone by 3 beta hydroxy steroid dehydrogenase. Next ones are a little more simpler to remember. They're all hydroxylase. So 21 beta hydroxylase. 11 beta hydroxylase. Okay, this corticosterone again can be converted to aldosterone, which is done by aldosterone synthase. So mind you, this is done by your zona glomerulosa, this outer part that we were talking about earlier. So this is done over here. Okay, so that is how aldosterone is released. But that is not what we're concerned with right now. What is happening in zona fasciculata? That is what we're more concerned with. So we'll come to that. So over there, what is different is, so anything that is going down is all beta. 3 beta, 21 beta, 11 beta. But now we are going to take it sideways. So that is done by 17 alpha. Alpha is going to come. Okay. So this progesterone and pregnenolone, so these can be converted to this side. So over here we have an alpha, that is 17 alpha. And again, simpler to remember, hydroxylase. Again, you can see it's not that difficult. They're all hydroxylase. Going down is beta, going sideways is alpha. Okay. So then what is the product again? It's going to be 17 alpha hydroxy pregnenolone. Same thing, but just 17 alpha form, hydroxy form. This can again do the same thing, that if on this you apply the same 3 beta hydroxysteroid dehydrogenase. Same thing. Okay, that's why I started with this side. This will give you same thing, 17 alpha hydroxy, but progesterone. Now onto this, if you put another 21 beta hydroxylase, it is gonna give you a 11 deoxycortisol. Okay, so that is one different, corticosterone, here cortisol. So on to this now, if we put a 11 beta hydroxylase, I will finally give you your cortisol. So these series of steps that we saw, where first a 17 alpha hydroxylation, then it goes down through your beta route to finally give cortisol. That is what happens in your zona fasciculata majorly. Now inside this also, cholesterol to pregnenolone, that takes place in your mitochondria. But all the beta related ones, not all, 3 beta and 21 beta. So these steps actually happen in your smooth endoplasmic reticulum. Okay, there's some confusion in certain textbooks. In Ganong, it is mentioned that 17 alpha hydroxylase is actually present in the mitochondria, but most of the textbooks and review articles that I've seen mention 17 alpha hydroxylase to be again in your endoplasmic reticulum actually. So even Harrison textbook in medicine and all is talking about that. So I'll just go for that now. So 17 alpha hydroxylase along with this 3 beta and 21 beta, all these steps are in the sarcoplasmic and the smooth endoplasmic reticulum. Rest of them all, the initial and the later stages, are happening in your mitochondria. Okay, so from this entrance questions and all come for you. So this is your cortisol. Now this cortisol is going into your circulation. The cortisol which is going into the circulation, what type of hormone is this? Steroid hormone. Steroid hormone are lipid soluble, not water soluble. So they cannot easily pass through your plasma. So they have to be bound with some sort of plasma protein. So generally we say that around 13 microgram of your cortisol is plasma protein bound, while around 0.5 microgram is existing in free form where it can act. Plasma protein bound, it is usually attached to something called transcortin or cortisol binding protein. Okay, which is a globulin. The cortisol binding globulin or transcortin, and it can also bind to albumin, but majorly it is to this. So now we have this 0.5 microgram free, which is finally going to act. Now, when you talk about excretion, again, this is not water soluble. It has to go through liver. Water soluble ones go through kidney. So how does the excretion take place? Excretion of this, it goes to your liver, where conjugation is going to happen. So in your liver, this gets conjugated. And the conjugated component Out of that, 25% is excreted in your bile. 75% goes out after conjugation in your urine. That is how it is finally excreted. So what do we have here so far? We have how your release occurred, the HPA axis, factors that regulate it. Then we talked about how the synthesis and secretion of your glucocorticoids are happening, all the steps that are essential over here. Then we briefly talked about how much is bound, how much is free, and how it is excreted and all that. Okay. Now, after this, I'm going to go to the functions and mechanism of action, which are very, 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 very important. Okay. So Cushing syndrome comes very often as an essay, so it's very essential for you to understand what are the various sub questions that come. One among that is your functions. Okay. So if you have any doubt with anything so far, please do let me know. Okay. So now your cortisol. I just put a cortisol looking like this. Cortisol is again lipid-soluble. I've been keeping on repeating this. Okay, so cortisol, which is lipid-soluble, will pass through the membrane easily and reaches inside. So inside, we have a particular type of receptor which is in the cytoplasm. So these receptors are usually attached to a particular substance, which is a heat shock protein. So it is HSP chaperone. It usually is attached to your heat shock protein and stays in the cytoplasm. Now, when cortisol is coming, it comes inside. And once it comes inside, then your chaperones are degraded and your corticosteroid, it binds to your receptor in the cytoplasm. Now, once it is bound, this receptor complex then goes to your nucleus. In the nucleus, in your genetic material, we have a particular area which is called a hormone response element. So what happens here is this has to 1 and 2. Like this, it has to dimerize. And how is it dimerizing? It's the same things that are happening. Two similar components are there. So it is a homodimer. So it dimerizes, the homodimer is formed, and that will initiate the transcription from the hormone response element. So a series of transcription will happen. That will start off all the functions. Now this acts in a different way also. Not only does it dimerize with the same sort of component, instead there can be another component which is called your activator protein 1. So instead of two similar looking components, now what is going to happen? One activator protein and one of this. So this is a heterodimer. When it attaches like this, it does not result in transcription. Instead, it results in transregression. So this is another way by which it reduces certain other functions. One example is how it reduces pro-inflammatory cytokine sinks. This is one of the reasons why it has anti-inflammatory effects. Okay, so what did cortisol do? It came in, bound to its receptor. Usually the receptor is attached to a chaperone. Okay, now that gets degraded. Now the receptor and the cortisol becomes a complex. They dimerize and result in transcription. But they can also form a heterodimer with an activator protein and results in transregression instead. Now we'll come to the various functions. So under functions, we'll be talking about one, the metabolic function. Metabolic function, what it does to carbohydrate, what it does to your lipid, what it does to your protein. Then we'll talk about your anti-inflammatory function. On top of that, there are certain other functions like permissive activity. So let's come to that. So when I was talking about the growth hormone, I had mentioned something about counter-regulatory hormones. On one side, insulin is there. On the opposite side, there are a group of hormones which are there, like growth hormone, glucagon, catecholamines, and your cortisol. Okay, so one side insulin, and the opposite side, we have these. So what does this do for carbohydrate metabolism? So your glucose, which is there, has to come in through your GLUT4 receptors. Now that glucose comes in, that is utilized for glycolysis. Once glycolysis happens, one of the end product of that is your acetyl CoA. And then it's going to go to your TCA cycle and give you energy. And will give you energy. So this is generally how it works in our body. You get glucose. From glucose, it goes into the cell and it is used for glycolysis. And then finally it goes to the TCA cycle and then you get energy. For this, you need insulin. Insulin, what does it do? Insulin acts to bring this GLUT4 receptor. Okay, this transporter is brought to the membrane so that glucose can come in. So for glucose to come in, we need insulin. Okay. So what does your cortisol do? One, it reduces the uptake of glucose. Two, the ability of insulin to be able to bring GLUT4 and then result in glucose coming in, that ability is also good. So this is what we call insulin resistance. Okay, so glucose is not coming in. Ability of insulin to be able to bring glucose in, that is also stopped. So now we're not getting energy from glucose, which means we need some other source for energy. So if glucose is now the next best source is fat. We don't go for protein immediately. Okay, so what will happen to fat? So for fat in adipose, so inside the adipose, we have your triglyceride. So hormone sensitive lipase is stimulated. So this is stimulated to result in triglyceride to be broken down inside adipose to free fatty acids. This is something cortisol does. Why did it do that? Because this free fatty acid is now gonna come and it is gonna go into the cell, gets converted to acetyl-CoA and gives you energy. So now the major source of energy is free fatty acid. That is why this was lipolytic. Lipid catabolism. Okay. Now beyond that, what happens to your proteins? So your proteins are formed from your amino acids, or proteins can be broken down into amino acids. Okay. And for this, you need amino acids to come into a cell. What does your cortisol do? One, it increases the catabolism, that is breakdown of proteins. Two, it inhibits the uptake of amino acids into general cells. Most of the cells, it inhibits the uptake. Okay, so this is also catabolic protein. Catabolic. This is also catabolic for lipids. I hope you are there with me till now. Okay. So why is all this done again? Because what was the initial thing that stimulates the release of your corticosteroids? One, stresses. Anytime your body goes to some sort of stress, pain, stress, and at that time, glucose is essential for major organs like the brain, for example. Your brain is something that can only rely on glucose. It cannot actually take supply from your protein and fat and all. It needs glucose. So in a sort of stress, you need to make sure that the brain receives glucose. The rest of the body can utilize fat and protein. So you need to make sure that your glucose does not go into all the cells. So insulin resistance should happen so that the glucose can go majorly to the major organs which are needing it. And then it tells all the other cells to start utilizing something else, like fat. Okay. And in major stress and all, you need to start breaking down protein also. Okay. So we are not done yet. So there is a small little organ over here, which is your liver. So the free fatty acid that I was talking about, this free fatty acid will go into liver also. Now remember over here, the amino acid uptake into cells are reduced, except in your liver. So somehow only into the liver amino acids are taken up more. Why is this? Both amino acid and free fatty acid, if it goes into the liver, it is going to give you glucose. This is what you call gluconeogenesis. Why did this happen? Because we needed more glucose to go outside, so that brain requires it. Okay, so not only did we make sure that glucose didn't come into cell, but we also produce more glucose from new sources. Okay. Now again, because amino acids came in, this is catabolic for proteins in general, except in your liver, because amino acids are coming here, it is gonna help in plasma protein synthesis and other proteins from the liver. So generally in protein, you have to remember cortisol is catabolic, except in your liver where it helps in production of stuff, protein. Okay, especially plasma proteins. This is because amino acid transport into the liver is happening. Why did that happen? For gluconeogenesis. Now, I'm going to tell you another point, which does not go sit well with rest of the thing I was talking about. All of this actually makes sense if you think about it. But one thing that doesn't sit well with what we're talking about, that is glucose gets converted to glycogen. And this is something your cortisol does, which is quite baffling if you think about it. Okay, if you wanted more glucose outside, shouldn't you actually break down the glycogen and give more glucose and give it outside? That is what you would have expected this to do. But cortisol, instead of doing that, made sure more glycogen is formed and stored. Why it did this is what he called permissive action. So this did it so that other hormones like glucagon or catecholamines can now come and break glycogen to give more glucose. Okay, so something should promote. If you stop insulin, or the activity of insulin is completely stopped, there is no glycogen forming storage. Okay, so what that does is cortisol does is, along with all this, it does only glycogen is stored, which is quite contrary to most of the other thing it does. This is so that now your other hormones like glucagon and your catecholamine can come and break it down to result in glycogenolysis. Okay, so there we have covered your metabolic. This is a very important question. Okay, what it does to carbohydrate, what it does to lipid, what it does to protein. I hope you understood it so far. If you have any doubt, please do let me know. Next, I'm going to your anti-inflammatory. This again can separately come as a short note. And the thing is, when students get this question, they do not have enough points. Most of the textbooks do not have enough points for you to, write an answer out of it. Okay, so I'll try to explain why it is anti-inflammatory. So remember, I was telling you here earlier, external glucocorticoid. Why are we giving external glucocorticoid? Usually we give external glucocorticoid when there's some sort of, inflammation in the body. A lot of inflammation, so autoimmune disease or some other sort of, inflammation that is chronically happening, you can give glucocorticoids for long to suppress that inflammation. Okay, so that is one function of your cortisol, which we take advantage of. So for that, you have to understand what is inflammation. Okay, so when there is a virus or any sort of cytokines which are released, or any sort of oxidants are there, anything that causes stress. Okay, so virus, cytokines, oxidant, any of these things like this. This usually goes and stimulates a particular component which is called NF kappa B, nuclear factor kappa B. Okay, so this is actually made up of two subunits. Okay, at your level, I don't think it's needed, P50 and P65, but this is made of different subunits. So this is NF kappa B. Okay, so when virus, cytokines, all this is there, this NF kappa B goes and acts as a transcription factor. Now, normally this NF kappa B is attached to another component, which is inhibitory in nature, I kappa B alpha. So as long as it is attached to it, NF kappa B doesn't act. But when all these signals are there, NF kappa B goes away from this and this goes into degradation through ubiquitin chaperone mediated. Okay, proteasome mediated degradation happens. So this is transcription. Transcription of what? What exactly does this do? Okay, so Now, let's take a blood vessel. We'll try to explain what are the things that happen during inflammation. These are the endothelial cells. Imagine a bacteria, a virus, whatever it is over here. Okay, so now this is there. Your antigen presenting cells come over here. Either this or there is some antigen antibody complex, like an autoimmunity and all. Okay, so anything, ultimately what is being released because of this, some sort of cytokine is released. Okay, or because of the presence of eosinophil, basophil and all that, certain other substances like histamine, bradykinin, mast cells and all, bradykinin, prostaglandin, all of these can get released. So the end result of all of these components is one, it results in vasodilation. So when there is vasodilation, there is increased blood flow. Two, it results in increased capillary permeability, which means from inside things can ooze out easily. Three, there are WBCs which are circulating in your blood. So they can migrate to this location based on the presence of the cytokines. And what do? They go start engulfing this. This is your phagocytosis. Okay, so first phagocytosis happens and then phagolysosome. Okay, so once this is engulfed inside, what will happen? Your lysosomes also start coming and they come and degranulate into this and start breaking it down. So that is phagolysosome formation. And that is how you destroy this. Okay, and on top of that, if it is a chronic, from your bone marrow. So in your bone, you have the bone marrow. From this bone marrow, your WBCs are produced, which will reach your bone. So these are various steps that happen during any sort of, inflammatory procedure. Okay. What exactly does your cortisol do now? Once you understand this, now we can understand what cortisol exactly does. So cortisol, what it does is, one, we already talked about it over here. Okay. By transregression, it decreases the pro-inflammatory cytokines. Okay. Two, cortisol will go bind to one particular subunit of NF-kappa B, your P65, and blocks its activity. So it no longer acts as a transcription factor. Two, it makes sure that the degradation of the inhibitory factor which is attached to it is not there. So it is existing more. Okay, the thing is, NF-kappa B, when it is detaching from your inhibitory component, it goes in X. So that inhibitory parts degradation is inhibited. So we have more inhibitory component. And as a result of this, what happens? As a result of this, from the bone marrow itself, your WBC production, which is happening, this step by itself is reduced. And if you look at it, there is a reduction in lymphocytes, eosinophil, and your basophil. The cortisol decreases your lymphocyte, eosinophil, and basophil. Okay. It decreases your pro-inflammatory cytokine release. We talked about it earlier. And when the eosinophils and basophils are reduced, the cysteamine, bradykinin, all of this release is also not there. So because of all that, what is happening? One, this vasodilation, this should happen. That does not take place. Capillary permeability, which is supposed to be increased, that is not happening. Okay. WBC is being produced from the bone marrow, that is reduced. Your migration of WBCs into the location, that is also reduced. Phagocytosis, that is also reduced. This is a very important point. Lysosomal membrane stability. Okay, so basically it stabilizes your lysosomal membrane. This is one of the most important function of cortisol in anti-inflammatory activity. Okay, so under anti-inflammatory activity, what do we have to talk about? One, it results in transregression through your activator protein forming a heterodimer. So pro-inflammatory cytokines are reduced. Then your NF-kappa B is kept inhibited by increasing the inhibitory component, by decreasing its degradation. And also it attaches to one component of NF-kappa B to again decreases the activity. And your cytokines are reduced, histamine, bradykinin, prostaglandins are reduced, because of which vasodilation does not take place, capillary permeability does not take place, migration of WBCs into the location of insult does not take place, phagocytosis is reduced, stabilization of lysosomal membrane is happening, and there is generally decrease in the lymphocytes and bases. On the other hand, you see that there is usually an increase in your neutrophil. There is going to be an increase in your RBC. There is going to be an increase in your platelet. That is why when Cushing syndrome, excess of glucocorticoids is there, because these are reduced, they are more prone to have infections. Because platelets are higher, your thromboembolic episodes and this embolic episodes are higher. Okay, now let's come to this. When excess is there, what is happening? Some sort of insulin resistant diabetes like state is there. Okay, protein becomes catabolic. So when excess of that is happening, means the muscle weakness will happen. Person will have weakness of the muscle and all. Again, fat again has a slight difference. You have peripheral fat and central fat. Okay, so it goes for peripheral fat more than your central. It's not that it goes for peripheral fat more, just that central fat production is slightly higher. But ultimately, what is happening? Peripheral fat is reduced more. So because of this, you start having more, the periphery is very thin, but the central parts become, big. So the proper buffalo hump, moon facies, all of these start developing. Okay, so this is essentially what Cushing syndrome is. So we talked about Cushing syndrome just by understanding all the mechanisms that are happening here. Okay, one small thing I want to end it with is your permissive activity. What is permissive activity? So the actions of your cortisol is such that it helps certain other hormones to now act better. So for glucagon and your catecholamines, their calorigenic activity, heat production that is done by them is helped by your cortisol. Also, the glycogen genolysis. For that, I told you, glycogen is formed with the help of this. Again, beyond that, this is a very important thing. Catecholamine. One of the major function of that through the alpha-1 receptor is vasoconstriction. So such pressor activity of catecholamines requires your cortisol. If cortisol is not at all there, pressor activity is going to be largely reduced. Okay, and on top of that, catecholamine mediated lipolytic activity and your bronchodilation. All of that also requires your cortisol. Okay, so this is what you have to talk about under permissive. So I think in short, I covered everything. So if you can, go through this every time you need to study, it'll be very helpful for you. Okay, if you have any doubt, please do let me know. Okay, so I suppose there's no, okay, doubts or such anything. Okay, all right then. Thank you. See you again next time.